In Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to our second lesson on genetics. In our last lesson, we learned the basics of genetics, the definition of a monohybrid cross, and how dominant genes affect the phenotypes of organisms. Today, we will expand our knowledge by studying dihybrid crosses. Are you ready? A monohybrid cross is the term that is used to describe the inheritance of a single characteristic in an organism. A dihybrid is an individual who is heterozygous at two different gene loci. We will discuss the concept of dihybrid in greater detail soon. But first, let us review some terminology that will be important for us to know in this lesson. Please write out the definition for the following terms: homozygous, heterozygous, dominant trait, and recessive trait.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. I hope that was a good way to review. These terms will be used often throughout this lesson, so let us review their definitions together. Homozygous refers to having identical alleles for a single trait. Heterozygous refers to having two different alleles for a single trait. A dominant allele is the allele expressed in a heterozygous organism. A recessive allele is the allele expressed only in the homozygous organism. If your definitions were similar to mine, well done. Now, let us continue discussing dihybrid crosses. A dihybrid cross describes a mating experiment between two organisms that are identically hybrid for two traits. A hybrid organism, as you know, is one that is heterozygous, which means that it carries two different alleles at a particular genetic position, or locus. Therefore, a dihybrid organism is one that is heterozygous at two different genetic loci. In our previous lesson, we learned that the scientist Gregor Mendel did a lot of work in order to understand genetics. In 1865, he performed dihybrid crosses on pea plants and discovered very important information about the laws of genetics. He formulated the law of independent assortment, which states that the inheritance of one trait is independent of the inheritance of another. In other words, one pair of alleles that control one feature segregates independently compared to another pair of alleles that control a different feature. Mendel began his experiments by crossing two homozygous parental organisms that differed with respect to two traits. An organism that is homozygous for a specific trait carries two identical alleles at a particular genetic position. Mendel crossed a pea plant that was homozygous and dominant for round and yellow seeds with a pea plant that was homozygous and recessive for wrinkled and green seeds. The cross can be represented in the following way. Round seeds, expressed as capital RR, and yellow seeds, expressed as capital YY, are crossed with wrinkled seeds, expressed as lowercase rr, and green seeds, expressed as lowercase yy. Organisms in the initial cross are called the parental or P generation. The offspring of the dominant and homozygous round and yellow seeds crossed with the recessive and homozygous wrinkled and green seeds is called the F1 generation. The offspring from this generation were all heterozygous plants with round yellow seeds, and the genotype capital R, lowercase r, capital Y, lowercase y. Next, Mendel crossed two plants that were both heterozygous for both features from the F1 generation. This step is the dihybrid cross. It is represented as RRYY crossed with RRYY. Mendel observed that the F2 offspring of his dihybrid cross had a 9:3 to 3:1 ratio. This generation produced 9 plants with round yellow seeds, 3 plants with round green seeds, 3 plants with wrinkled yellow seeds, and 1 plant with wrinkled green seeds. From his experiment, Mendel observed that the pairs of traits in the parental generation sorted independently from one another, from one generation to the next. Let us take part in an activity to review what we just covered. Please complete the following statements.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. That should have been a fairly simple activity for you to complete. Let us go over the correct answers. A dihybrid cross describes a mating experiment between two organisms that are identically hybrid for two traits. Mendel's law of independent assortment states that the inheritance of one trait is independent of the inheritance of another. The dihybrid ratio of the F2 offspring is 9:3 to 3:1. Did you write down the same answers? If so, you are certainly paying attention today. Great job. As we have learned, there are clearly more possibilities when we are dealing with dihybrid crosses than with monohybrid crosses. To review, the offspring may be homozygous for both features, which is represented by RRYY. The offspring can be heterozygous for both features, which is represented by RRYY. The offspring can also be heterozygous for one feature and not the other, which is represented by RRYY or RRYY. If we refer to the diagram of the Punnett square for a dihybrid cross, we can get a clearer idea of how the cross occurs. Take a moment to study this diagram. It is important to note that alleles are not always simply dominant or recessive. Sometimes alleles can be codominant, which means that neither one dominates the other in the heterozygote. In the example of a snapdragon, the flower color is controlled by a single gene that has two alleles. If both of the alleles are dominant, then the petals are red. If both are recessive, the petals are white. However, if one allele is dominant while the other is recessive, what do you think happens then? It would be reasonable to expect that the flower petals would be red, since we have learned that dominant genes override recessive genes. But you might be interested to learn that both alleles still express themselves. Red pigment is produced in the leaves, as is white pigment. The result is that the petals are actually pink. If a red-flowered snapdragon is crossbred with a white-flowered snapdragon, all of their offspring will be heterozygous and have pink flowers. If two of these heterozygotes are crossed, we get the same genotype ratio as with any other monohybrid cross. But instead of a 3:1 phenotype ratio, it is 1 red, 2 pink, 1 white. We have reached the end of our lesson. Today, we have learned about dihybrid crosses. Until next lesson, thank you, teacher. Thank you, students.